All right, everyone, we're going to take a journey through the digestive tract. Not like literally like magic school bus style, but we are going to at least talk through all the major structures and then what we're going to secrete into it and what secretes into that pathway. Now, I do want to split it up because it's a lot of information. So in this video, we're going to focus on the upper part of the digestive tract. So we're going to go mouth, through the pharynx, so throat, down the esophagus, to the stomach, and we'll talk about all of the cool things that happen along that way. So first we'll start with where mastication occurs, where we put food. So the process of ingestion is when we put food in our mouth. It is considered a conscious process, even though, let's face it, we've all done the thing with like half a bag of potato chips going, I don't remember eating all of that. So we're gonna start all the way at the oral cavity. So with that oral cavity, there's a lot of things going on. One being mastication. It's a physical process of chewing food using your teeth. The tongue's also moving things around to form a bolus so we can push it to the back of the mouth when we're ready to swallow. And there are also secretions coming in. So we have secretions coming in from different structures. The tongue actually does secrete. So the tongue will secrete lipase. And if you see ACE in the end of something, that means it's an enzyme. So ASE is telling you that you have an enzyme here. Lipase, to help you out, is telling you it's a lipid, so it breaks down lipid. We'd specifically call it a lingual lipase because lingual meaning tongue, it's where it's coming from. Because later, when we get to the stomach, there's going to be a gastric lipase, one from the stomach. Or if we get to the pancreas, we'll have pancreatic lipase. So they're just telling you which structure is secreting it. So the tongue will secrete that lipase into the oral cavity. So we can start breaking down fats in the mouth. The other major nutrient that we can start breaking down in the mouth is going to be carbohydrates. And that's going to be broken down using amylase, so a different enzyme. Now that'll come from salivary glands. There are actually 3 different salivary glands. So we have sublingual, submandibular, and parotid. All of these are going to secrete into the oral cavity, but they don't all secrete the same thing. So I did mention amylase. Amylase will be secreted by these 2, but not by this one. So this one here and this one here will have amylase. So I'm going to shorten it. So we'll have amylase from those. And that's the only thing coming from parotid. So I guess I can fit the whole thing. So we're going to have salivary amylase because what we're secreting is saliva. Here we'll have amylase, but we'll also have mucus and buffers because we do sometimes consume things that are acidic. So we want to help neutralize that environment inside of the mouth. We'll secrete buffers for any of those acidic foods. It helps protect the enamel on your teeth. The sublingual gland will also have mucus and buffers, just like what we see with the submandibular. So this is a trick that I use. It's like all about pairing. See where your relationships are. So these 2 duplicate here. This one here duplicates there. So that's kind of my pairing. The other thing that I help myself with is there's an L here. The thing that gets secreted from here that's different, that's unique, also starts with an L. And that's lysozyme. So lysozyme is an antibacterial. Seems like a really good thing to put in your mouth, right? Because let's face it, the 5-second rule is not a real thing. Hopefully I did not just ruin that. Or have you ever done the thing where the pizza stayed out all night and you're like, yeah, I'm sure that's fine. I can eat that. No, but hey, you have an antibacterial. Don't make that your primary plan, but it can help you out. So we have an antibacterial lysozyme secreted by the sublingual one of the salivary glands. So we have salivary glands secreting saliva into the oral cavity. So if we want to take a look at what those look like, if you're trying to find them, the parotid's the big one here. So if you're looking at the picture, whoa, I gotta find my picture here. That's the one over here by the ear. So the parotid, if you consume stuff like a lemon head, something really sour, you can almost feel it working as it's trying to secrete things to help you digest that sugary substance that you just put in your mouth. So we're breaking down complex carbs with the secretions, and so they're like working, you can feel it kind of tightening up in that jaw. Then we have sublingual, with a name like that, sub below, lingual tongue. This is the one right underneath the tongue, like right here. Below that one, so the one further down, that's submandibular. So you can see the mandible's missing in this image. They cut it off so you can actually see underneath where that submandibular salivary gland will be. So that's what's going on inside of that oral cavity as far as what are we putting in there. But now what about the mechanical process? So this is our chemical processing. With the mechanical processing, we're going to be using the teeth. Now with teeth, we do have baby teeth, and then we develop adult teeth, and with the adult teeth, they all have different names. Some advice on how to remember them is use what you have. So if you have a question on a test and it asks how many incisors are there, well, the incisors are the flat ones in the front. So take your tongue and actually feel around, and you'll feel those ones that are flat for clipping at the front. And so you'll have, if you can kind of feel it, 4 at the top and 4 at the bottom. Then you move your tongue next to those two, you'll feel a pointier one. That's a canine. So then the canine, or cuspid, has one single point. Now if you look in our picture over there, after the pointy one, do you see how these two right next to that have 2 points? Those are called bicuspids. And then the ones that are flat for grinding in the back, there's a row right here of 3. So we have 3 on this side, 3 on that side, 3 here, and 3 here. Those are the molars. Now this is where you have individuality, because some people don't have molars. Some had them removed, and it's actually an example of evolution currently happening in humans, where with every generation we're seeing less and less molars actually ever forming. Long, fun story, but I have that in the original lecture videos. With the mastication process, you're chewing it up, you're increasing surface area so that these chemicals can then get to that molecule and help break it apart. You then have mucus to help kind of add some binding, and you can make it into a ball. That ball is called a bolus. That is what you swallow. So what will happen is when we're ready to swallow, the tongue will push it to the top, to the hard palate, which is, if you're looking here, this is the front part here. The hard palate's made out of bone, and then you have a soft palate in the back. So you're gonna push it up. That's at burst, and it's the voluntary part of swallowing. Once you then push it back in towards the pharynx, then it's gonna become involuntary in the pharyngeal phase of swallowing. Once it hits in that area, we no longer can like stop it in reverse direction, because we're going to lose our ability to breathe. So smooth muscle in the pharynx will automatically start pushing it down towards the esophagus. And so then it'll be our third phase of swallowing, will be esophageal. So we'll have that conscious voluntary phase first, tongue pushing up, then we'll start to push back, then we'll go pharyngeal phase. We're gonna work our way down the pharynx, which is the throat, shared passageway, and then we'll move into the pharynx. So I probably should write some of this down. So we have oral cavity, we'll go pharynx. Pharynx is a shared passageway with the respiratory system. So we want to get the food out of there very quickly, so a smooth muscle is going to push it through there. You won't push it into the airway because there's an epiglottis that blocks that airway when we're swallowing. And then we're going to go through a ring of muscle at the top of the esophagus, so we call that a superior esophageal sphincter, because it's superior for the top, and then esophageal for the esophagus. And then from there, we're going to go esophagus. Now, we haven't talked yet about what is lining all of this. All of this so far is going to be lined with a stratified squamous epithelium. So stratified squamous epithelium, because you have a lot of rubbing here, and it is non-keratinized. So non-keratinized, they're all abbreviated, hopefully this makes sense. So non-keratinized stratified squamous epithelium. You have a lot of rubbing here, and the cells are all alive. You're going to have mucus there to help kind of keep them moistened and keep them alive. You don't have them dead and keratinized like you do on the skin where they're dry. And so we have a lot of rubbing here. After the esophagus, that's when we can start doing exchange and absorbing things. So that's when we get to the stomach, that we will shift and change our look as far as the wall goes. All right, so esophagus, major events happening here is going to be pushing the food forward. Well, actually we have 2 major ones there. You can push it forward and you can chunk it up into smaller pieces. You ever swallow too much stuff at once? Not the best plan, but you have a backup plan. If it's too big and you're like, oh, that's a lot, your esophagus can then segment it. And so how this will work is our esophagus, and I just lost my image. So the esophagus tube here, it's in the mediastinum, so it's between the pleural cavity, so between the lungs, behind the pericardial cavity, so behind the heart. So we're hanging out over here in front of the airway. And that tube can actually squeeze on itself because there's smooth muscle there. And so let's say you swallow a large chunk of food, the wall itself can actually pinch and help you separate those 2 little balls from each other. And then you can move that one forward. And so then you kind of get some separation. And we're going to create segments of food. So that is a process called segmentation. So we'll do segmentation. And then we also will do what is called peristalsis, where we're going to push in on the walls behind it to kind of help propel it forward. And so those are the major events kind of happening in that esophagus as we work our way down. After the esophagus, we're going to have another ring of muscle. We can call it the inferior or lower esophageal sphincter. Or you can also call it the cardiac sphincter, because the top part of the stomach is called the cardia. Kind of weird. We'll come back to that. So this right here is the one that you might be familiar with if you have heartburn issues. So our cardiac sphincter is there to prevent acids and foods from going from the stomach back up that esophagus. And so if it's weaker, that's what we kind of would consider heartburn. So cardiac sphincter, lower esophageal sphincter, or esophageal gastric sphincter, because you're between the esophagus and the gastric. All right, so then that'll get us to the stomach. Again, a lot of cool things happening here. But let's recap a little bit. So here we had some mechanical processing. Here we had mechanical processing with those little segmentations. We have chemical processing starting here, but it can work all the way down. Then we get to the stomach. That's when we're going to get more chemicals added in. And so with the stomach, you're going to have 3 cells that are going to line what we call our gastric glands. So with the stomach, there are these big pits in the wall, increased surface area, we have folding there, that have cells that have different jobs. One is going to be hormonal job, another is going to be an enzyme job, and another one is primarily an acid function. So let's talk through each of those. Let's start with the parietal cell first. So we have a parietal cell, and so we're going to draw a parietal cell real quick. This does a reaction that you're already really familiar with, hopefully, and it's a really common reaction with our main byproducts in the body from cellular respiration. So water and our cellular respiration makes CO2 as a byproduct. So water and CO2 combine because there is something called carbonic anhydrase. So that's an enzyme sitting inside of these cells. And it will make carbonic acid. So that's carbonic acid. Carbonic acid splits up into hydrogen ions and bicarbonate ions. Bicarb is a lot of times in our blood to help buffer the blood to make sure it stays at a neutral pH. This we want to go put in our blood. So let's say this part over here is going to be our bloodstream. So there's a blood vessel. We're going to take that bicarb and we're going to stick it in blood. Now, if you're going to do that, we do want to exchange it for something else that has the same charge. So what we'll end up exchanging for is going to actually pass right along through, and that's chloride. We're going to do what's called a chloride shift. So we're going to pass the chloride this way. We will also pass the hydrogen into this side meaning the stomach side. So here we're passing it to the stomach, and so what we just handed the stomach is an extremely strong acid, hydrochloric acid. So we can lower our pH significantly in the stomach. Now, a couple of cool things happen with that lower pH. One is it helps denature the proteins that you've ingested, so it helps break down food. Another thing is it will be useful to have an acidic environment to activate a protein digester, because notice so far we don't have anything that can break down proteins. The protein digester actually comes from a different cell. And so that next cell is a chief cell. And so here's our chief cell. And this will make pepsinogen. You cannot make a protein digester inside of your cells that is functional, because most of you is protein. Your structure component that you would use to save this and store it inside of the cell would be made out of protein. It would be me like making a cage out of cookies to keep my kids in place. They're gonna chew through it and then freedom. So the pepsinogen, if it was active, would start breaking down the cell itself, and you would literally eat yourself. So we'll secrete it as pepsinogen, and then it hits the acid and it turns active. And we lose that gin part. Gin is telling you it's inactive. Pepsin means it's active. And so because of the acid in the stomach, we can get pepsin, the functional version, to break down proteins. Now, because an acidic environment does denature proteins, these enzymes that you released up here that we're working along the way, they're going to get denatured because acidic environment will change how they look and it won't be functional anymore. So that's another interesting thing there. It's like you will actually stop your carbohydrate digestion here. You will secrete another lipase. So you will still be able to digest lipids. This one just works in the acid environment. And again, we call this one gastric lipase because this one was our salivary lipase, or sorry, lingual lipase. So here we're going to release a protein digester and a lipid digester. We won't get another digester for carbohydrates until we move into the small intestine. And speaking of which, we should probably mention that real quick too. So after the stomach, there is a ring of muscle on the other end called the pyloric sphincter. This is the barrier between your stomach and your intestines. You can do some absorption at the stomach, which is why you have a change in what's lining it. So starting here and the rest of the way down, I guess we can just say this is muscle, but it would have this liner here. It's not very big, but I'll include it up there. So here we can start to do some absorption. Now, not a lot. Some things like some drugs, alcohol can be digested or uploaded from the stomach. So this is going to be simple columnar epithelium. And so that allows you to do absorption. Okay, now there is one more cell, because I did mention there would be 3 of them. This one, however, does not secrete into the stomach. All right, so we have this G cell here. The G cell is not going to secrete into the stomach. Instead, it's actually going to be secreting hormones. And if it's secreting a hormone, it's going to send it into blood. So gastrin is the name of the hormone, and that's going into the bloodstream. So gastrin is interesting because it's going to come back and talk to the walls of the stomach itself. The job of gastrin is to promote the gastric phase of your your stomach activity. So the stomach activity is going to be split into 3 parts. The first part of stomach activity is based on what you see, think. So it's signaling from the brain. It's called the cephalic phase because cephalad means head. So it's your head seeing food and thinking of food and picturing food, and now you're salivating and you're getting ready for food, and we're going to start to secrete and wait for the food to get there. Once the food gets there, and the stomach can tell from a variety of ways, chemical signaling because you have chemoreceptors on the surface, or stretching, so mechanoreceptors built into the wall. And so once it knows food's there, it's going to secrete that gastrin to kind of circle back and say, yeah, food's here. Now we can start mixing stuff up. We can secrete a little bit more. Then we pass food into the small intestine, and the intestines will have a separate set of hormones that it will use to talk back to the stomach for intestinal phase. Say, okay, you've handed your food off. You can stop doing what you were doing. I got this now. So you do have some signaling happening from the organs themselves, which is kind of cool. And so this is kind of some major events happening here. Now there is one thing I did mention where it's like, okay, you have a cell in charge of pH, a cell in charge of enzymes, and a cell in charge of hormones. For the most part, I did say there's going to be one oddball. There is one oddball. It comes from the parietal cell into the stomach, and that's going to be your intrinsic factor. An intrinsic factor is actually going to be used in the intestines to help absorb vitamin B12. So it has a helper or support role for absorption of something that we do need, and it has a harder time passing through the wall, so there's a little helper secreted from that one. Okay, now a couple of anatomy things on the stomach, and then we're pretty much good on this video. So looking at the stomach here, we have that esophagus coming in. You can see there is just a little ring of muscle here. That's going to be that lower esophageal sphincter. Then if you notice, we have a greater curvature. That's the big arch. Then over here we have a lesser curvature. On the inside of the stomach, we have a lot of folds. Those are called rugae. It allows our stomach to expand to like a liter or a liter and a half. Like next time you picture a 2 liter of pop, most of that can fit in your stomach, which is kind of weird. Don't try it, but it can get that big. So we have those rugae to increase that surface area so we could expand if we need to. And then at the bottom here, that's that pyloric sphincter, which leads us into the small intestine. Pyloric sphincter helps control how fast we hand things off between the stomach and the small intestine, because the small intestine, specifically the first 2 segments of it, or really the middle segment of the small intestine, is where most of our absorption takes place. So we want to make sure to give it plenty of time. So releasing things a little bit at a time from that stomach and give the intestines time to process it, add some more enzymes to do some more chemical breakdown before adding more from the stomach. So you don't want to just open the gate and say, okay, all of you out. You release a little at a time. All right. Hopefully it was helpful to kind of see it all put together, at least from the upper half of the digestive tract. Thanks for watching.